In this video, I'm going to talk about enzyme kinetics and so enzyme kinetics and receptor binding kinetics. So I will start with enzyme kinetics. So I talked about in previous videos how enzymes are catalysts. So their function is to catalyze reactions, which, if we remember from our thermodynamics, it reduces the activation energy. So if you have a catalyst like an enzyme, or it could be some other thing in inorganic, well, actually in organic chemistry, a lot of times you use transition metals. So transition metals. Um, but in biochemistry, we are going to be talking almost exclusively about enzymes. And so, like I said before, what happens in an enzyme catalysis is it reduces the activation energy. So what's happening is it's stabilizing the transition state. So this is from a paper that talks about the enzyme that turns corosmate into prephenate. I've only ever seen that written. So anyway, what's happening is it's breaking this oxygen bond here and binding this to right there. And so you see when it's in the transition state, when it's sitting in the active site of the enzyme, it's actually in a conformation like this, where we have some partial bonds going on right here, and this bond is strained, and so that bond wants to break, and this right here is in a favorable position for making a bond, and so we can sort of switch this over here and turn it into this species here. And so down here, this is just sort of a schematic sort of picture of it, where we have a substrate, and so kind of the canonical way of showing a substrate is it's one thing, and we want to split it into two, and so we're breaking a bond right there, and it turns it into two things. But enzymes can also do the other thing. It can take two things and turn it into one, or it can do this, which is just changing one molecule into a different molecule without adding or subtracting any other constituents to this molecule. And so a way we can schematize this is also in what are called Cleland schemes. And so if we have our enzyme here, we can have A bond first, and then B bond second, and then whatever catalysis is happening right here produces a product. It produces these two products, and one is released first, and the other one is released second. And we can sort of do that in opposite directions, so we can have A or B bind first. We see this kind of thing happen quite often when we're talking about an enzyme that phosphorylates something, and so it will have the substrate that it wants to phosphorylate, and then it'll have ATP. So both of these things have to bind. And so it might be the case that ATP will bind first, and then that opens it up for the substrate to bind. Or it might be the other way around, where we have our substrate bind, then that opens the enzyme up for ATP to bind. And so we can sort of schematize our reaction in this way. All right, so let's talk a little bit about the actual proteins doing this catalysis. So the thing that we want to talk about is, at first, is what's called the active site. And that's actually the part of the protein that you can see right here, where the substrate, which is the molecule that's being catalyzed, binds to the protein. And so this has some amino acids colored in green and red, and then we can see down here our substrate. And you can see here that there's hydrogen bonding going on, and so there are these amino acids in the active site, sticking into the active site, that that accommodate the substrate. And in fact, as I said earlier, it accommodates the transition state of the substrate. And so down here, I just have shown so chirality matters when fitting into the active site. So you can see here, this shows one where we have a nice ionic bond here, nice ionic bond here. We have some van der Waals forces here, and so this is fitting nicely into there. But if we use the other version of it, now we can form these ionic bonds here, but now all of a sudden we have some steric clashes going on here. And so when we're fitting molecules into the active site, it matters which one of the enantiomers, the enantiomers, are being used. And so, for instance, that's why in almost all life, well, all life on Earth that I know about, we are very specific for the enantiomers we use. And for instance, we use D-glucose. We always use D-glucose instead of the L-glucose, and we always use L-amino acids. I think I said in the amino acid video that there are a few, just a handful of notable exceptions in bacteria that use just a few D amino acids for very specific purposes, but that's the exception to the rule that will get pointed out if you ever come across it. And so anyway, like I was saying, it matters about the chirality. And so we want to fit our substrate into the active site. And so over here we just have some ideas about the active site. And so up here it's just talking about regulatory mechanisms. And so we can have our active site, but we could have some other molecule that binds somewhere else on the protein, and that might affect the active site. So that's sort of what I was talking about up here, where we have the substrate and the ATP, or it could be some other thing. If you might need one of those to bind in this site here in order for the active site to open up, or you can actually have it the other way around, where if you start having something bind here, then it actually closes the active site. And that actually you see quite often, because that's how a lot of metabolism is regulated. That would be called a negative feedback mechanism. And so what happens, and we'll talk about this quite a bit once we get more into metabolism, when you catalyze something with your enzyme, the product that comes out might bind here and inhibit this. And so if you start making too much product, then your enzyme becomes inhibited, so you stop making product, and that's why it's a negative feedback. It's it's sort of like your thermostat. Your furnace creates heat, and it's the heat itself that turns the furnace off. So that's uh, that's kind of how this allosteric thing can work. Like I said, there's also you can have allosteric activators, which turn a protein on, which turn a enzyme on, and quite often that's ATP. So that's often what you find. This is just talking about the catalytic function, which is what we're talking about. Uh, so we want to look for the cleft, which is the place where the protein can fit into the enzyme. Geometric analysis, so that's kind of more of the same thing. Flexibility is a big idea in enzyme kinetics. That's how like this allosteric thing works, and substrate binding causes conformational changes. Uh, Conservation, and so uh, we find in enzymology that there are a lot of different enzymes that sort of get repurposed for other things, and so you'll hear if you take biochemistry the CDK cyclin dependent kinase uh, enzymes, and you'll probably hear about the cytochrome P enzymes. So these are enzymes that regulate the cell cycle and cell division, and these are enzymes that largely um, break down things like toxins and other molecules that end up in your body. So we have multiple of these CDK8s. Well, I just call it CDK8 because that's the one that I worked with in my graduate work. So there are CDK molecules, of which the one I was working with, CDK8, was just one example of a CDK molecule. And cytochrome P, we'll actually run into some cytochrome cytochrome P-like molecules. For instance, when we're biosynthesizing vitamin A, so there are cytochrome P-like molecules. So, like I said, cytochrome P, uh, one you'll often hear about when talking about toxic substances, and alcohol in particular, is the cytochrome P450. Uh, but this there's this conserved sort of sequence that gets repurposed into other molecules, and so we might repurpose it into something that that is used in the biosynthesis of vitamin A. And so that's, just kind of because of evolution. Uh, Evolution isn't isn't something that just creates things out of nothing. It sort of takes something that already exists and can repurpose it into new things. And so we can have, some other protein, and there might be a gene duplication, and then we repurpose it into we repurpose one of those genes into a new gene. Anyway, so we want to talk about surface characteristics. So that has a lot to do with interactions with other proteins and things like that, and interaction energy, which is just like the energy of. Uh, so that's thermodynamically how much, say, a substrate might want to actually enter your protein. All right, so we talked about chirality. Right here, I have just an instance of a, I don't want to say common. It's seen though, this this nucleophile base acid sort of motif going on inside the active site. I think this is what we'll see in things like trypsin, which is an enzyme in the stomach that breaks down proteins, and so you can see how that would work if this was a peptide bond. And so, like I said, this is just an example of one of many types of catalytic mechanisms. And I've mentioned in previous videos before, we see that we're using a ketone here. A lot of biochemistry is ketone chemistry, or carbonyl chemistry, which is things like ketones and aldehydes and other things where it's a carbon double bond oxygen, esters. And so anyway, this just shows our base, which I think is quite often histidine, and our nucleophile, which is quite often serine, and so this would be an oxygen, this would be an alcohol group, and our histidine can extract that proton off of it, turning this into a negatively charged oxygen, which will attack the partial positive on the carbon here, kicking the electrons up to the oxygen. And then this oxygen will have the electrons come back down, reforming that double bond. And I mean, sometimes it might go to here and kick this back off, but the active site will probably hold this intermediate in a position where having this get kicked off is more favorable. And so we removed, so this shows the R grabbing that hydrogen off of our base, which, like I said, is often histidine. And so this leaves the active site, this leaves the enzyme, and now we want to bind something else to our substrate here. And so our base, because I mean, this is still bound to our enzyme here. We have this this intermediate here that has this covalent intermediate, and so now our base can deprotonate. Say maybe this is another oxygen or another alcohol group or something like that, and that will come and bind here. Then this bond breaks while grabbing the hydrogen off of there, and we're back reconstituting our active site of the protein. See, this is the same as this. And that's sort of the general definition of a catalyst, is that it's something that speeds up a reaction without getting used up itself. And so we need our active site to be back in the exact same way that it was when we began the reaction over here. Down here, I just have I thought this is a cool image, and it's from a paper. The link to it should be in the description, and it's just an enzyme that has this large conformational change when this substrate binds to it. And so you see this what they're calling the lid sort of folds over and closes up on top of the substrate when it goes into the enzyme. And so that's fairly common for these large conformational changes to occur when the substrate binds to the enzyme. Down here is so this is kinetics, but it's actually more thermodynamics, but it's important enough that it needs to be somewhere, so I'm putting it in this video. And so this is sort of the reason why we use ATP. So I think when a lot of times when you take like a biology 101 class, they'll say ATP is the energy, and you think of ATP as this molecule, and it just sort of shoots a laser over at a bond and causes that bond to break or something. But that's not what happens. What uh what happened? So we see here our ATP, negative delta G. That's a favorable reaction, goes to ADP, diphosphate. We have two phosphates on it. It requires energy to add a phosphate onto it. This is doing this reaction here is the reason that we eat. So the energy that we get from things like carbs and fats and even proteins, the energy that we get from these foods, the calories, which are a unit of energy, is used to do this, because then we have this energy here that we can use. And the way that that's used is what's called a coupled reaction. So it shows here we have two molecules, two substrates, and to form them into one molecule, to bond them together, takes energy. So I talked about in the thermodynamics video, we look at, when we're doing thermodynamics, we look at things from the point of view of the system. The system is this. The system are the molecules. And so energy is going into the system, and that's why it's plus. That can always be a little bit confusing, because it's like, well, plus, isn't that the energy that we're getting out of this? Aren't we getting plus energy out of it? But this is from the point of view of the system. We're putting energy into the system. So this here is higher energy. So if we have our reaction here and our energy here, it's saying that our reactants are up here, then our product is up here. And so this is at a higher energy. But this isn't going to happen on its own, because, well, thermodynamically, it's not a spontaneous reaction. Positive delta G is not spontaneous. It's not thermodynamically favorable. The universe likes to be in low energy states. And so what biology does is it adds this ATP into our system. And the ATP, when the ATP going to ADP here, has this negative delta G. And so if we do this reaction at the same time as this one, all together, then we end up with a net negative delta G. And so by coupling these reactions, we're getting ourselves down to a net negative delta G, which makes this thermodynamically favorable. And so we'll run into this a lot, because there are a lot of enzymatic reactions that require ATP, because it's a reaction that is energetically or thermodynamically unfavorable, and so we use ATP to perform those reactions. And that's sort of the whole point of biology, or I mean, from the biological point, it doesn't have any sort of philosophical relevance to the meaning of our lives. But in biology, what matters is that we are increasing the energy. we are putting energy into ATP for the most part, so that we can use that energy to do other things. So I talked about in the thermodynamics video that equilibrium is so if we have this, then our delta G here is at equilibrium. We are always trying to keep ourselves further up here in the equilibrium. This is where the delta G was 0. So we're always trying to keep ourselves outside of equilibrium, because going from here down to equilibrium is how we extract energy. Anyway, this is turning into kind of a long tangent. Let's get back to kinetics. All right, now we reach the mathy part of kinetics. So kinetics is we can sort of draw it like this, where we have our enzyme here and our substrate, and the first step is they come together, and so now we have our substrate bound to our enzyme, and then the catalysis inside the enzyme happens. And sometimes you'll even see this written where there's another step where it goes to EP before then it goes to this, but this just shows it as the product being ejected as soon as it has catalyzed the reaction. So these K's here are the rate constants of these reactions. So this is the rate constant of the enzyme and substrate coming together. This is the rate constant of them coming apart without the catalysis going on. And so this K2 is the rate at which the catalysis occurs when we have our substrate bound to the enzyme. And you can also have it where enzyme and product come together, and then you actually catalyze it in reverse to form the enzyme and substrate. And so, I mean, that can happen. In biochemistry, there are often ways of sort of, as soon as this forms, we do something else with it so that this doesn't happen. So, for instance, if we're in a pathway, or like a biochemistry pathway, say glycolysis or something, which we'll get into in sort of a lot of detail in a later video. And so what'll happen is this product is some intermediate step in a multi-step biochemical pathway. And so some other enzyme will come and will actually use this product as its substrate. And so those will come together, and the reactions will continue going on. And so part of kinetics, well, a big part of kinetics is trying to figure out what these, well, we're trying to figure out sort of how much the enzyme and substrate come together and how fast this step goes. So we're looking for this, and we're looking for sort of the difference between K1 and K negative 1. So which is sort of the affinity of the substrate for the enzyme, which well, let's just go right down to it. So this is the Michaelis-Menten equation, and what we're trying to do here is find out what this well, we're trying to find out the V max and the Km for the most part. So what we do in a steady state enzyme kinetics assay is we we put our substrate at some given concentration, and our enzyme at some given concentration, both of which we know, and uh, we measure the formation of product. Uh, well, you can also measure the loss of substrate. Usually, when graphs like this are shown, it's looking at the formation of product in time. And so as soon as we add our substrate and enzyme together, we start getting product forming at this. Uh, well, at first it's a steady rate. As substrate starts being depleted, the rate is going to go down. But we're interested in what the reaction is doing at a given substrate concentration. And so that's why we pay attention to this linear part here at the beginning. And what we do is we take we do this at say one one substrate concentration, then we might increase to a different substrate concentration, then a different substrate concentration, and then we try to get this at multiple different substrate concentrations. And what you get then is a graph that looks like this. And so this down here, this isn't time anymore. This is substrate concentration. So we did an experiment at this. So this is our first substrate concentration. Then maybe we did it right here, and then we did it right here. And you always want to group it around uh, where where it's still going up here, and then you might want to get some concentrations out here just to see where it's leveling off, because that's where we get our V max here. And we saw in our Michaelis-Menten equation, V max is one of the things we want. So we know what the substrate concentrations are because those are things we are controlling. And we keep our enzyme concentration steady for all of these different assays. And then we can get a curve like this, and the other thing that we learn from this curve is what's called the Km, which up here is the other thing that we are trying to look for. And the Km is the concentration at which we are at half of our max. And so up here is where it'll max out, and no matter how much more concentration, how much more of the substrate you add beyond that, you're not going to get any more. You our enzyme becomes saturated, and the rate at which our substrate enters the enzyme active site is faster than you actually form products. And so this step becomes the well, this step is sort of saturated. This becomes the rate limiting step. And so when you reach that V max, that's how fast your catalysis is going. That's what this kcat is. So we take our V max and we just divide it by the total enzyme concentration, and that gives us this kcat, which is called the turnover rate. And so that's what this reaction is here. The Km is looking at this one. So Km is telling us sort of how much this is happening, how well the substrate actually binds to our enzyme. And so we see here that we use the half half V max. And what we find is that if our Km is smaller, so if Km was, say, here instead, then that means it's binding tighter, because that means that it had to get up to half before, earlier, or at a lower concentration than it did for this one. And so if you're at your half, if you're at half max, half of your V max at a lower concentration, then this is saturating your enzyme much quicker, because now uh, you're getting your rate limited much sooner, because the substrate is binding to the enzyme quicker. That's why you can reach your V max sooner. Um, and so that's how those are the components of the Michaelis-Menten equation. Um, one analogy I like to think about for this is uh, is is kind of a silly one, and it's just sort of, if you think about, if you are in a room and you're punching your fists sort of randomly around, and there's only one other person in the room, that the odds that you're going to hit that person is much smaller. And we can even say that you are blindfolded and they are blindfolded, and you're both just wandering around aimlessly. But still, uh, if you're in a room and they're there just wandering around aimlessly, you're not going to hit them that often. But now, if we add a bunch of other blindfolded people into the room, which is sort of like the concentration going up. So we got our blindfolds here. Uh, you're gonna you're gonna hit somebody a lot more often. Each one of your punches is going to end up connecting with somebody, and that's what we see here. Is as we increase the concentration of our substrate, which is kind of like the other people in the room. You are the enzyme. They are substrate molecules. So you see, if you increase the substrate, the chances that it's going to run into an enzyme molecule and become catalyzed increases. And so it increases up and up and up. But then we reach this V max here, and we can keep adding more substrate, but it's not, you're not getting more catalysis. And that would be like if we add even more blindfolded people into our room and it starts getting very cramped. So these people are all blindfolded. What happens is, now there are so many people that when you throw a punch, you might hit somebody every time, but now you are limited by how fast you can actually throw your punches. So the number of times a person is hit is limited by the number of punches that you have. And so that's what happens when you are at V max, is your, so this is kind of like the punching rate here. Uh, and so this is sort of the people wandering blindfolded about the room. This is your punching rate, how fast you're actually punching. And so if you have too much substrate, if there are too many people, then the rate at which you punch is actually going to limit how many people or how often you hit somebody, rather than if you have just a single person, uh, then you know you're blind walking around. How often you two actually run into each other in the room is what's actually going to be rate limiting in how often one of your punches connects with somebody. And so that's my silly little analogy here for thinking about enzyme kinetics. Down here, we can use our kcat, and so the kcat, which if you remember is sort of equivalent to your Vmax, which is when you were at at your highest there. And so we can divide our kcat by our km, and that gives us the catalytic efficiency. And so you can see how if we have kcat over km, we said that if km goes lower, then it's binding tighter. We said that if kcat goes higher, then then we're forming product faster. And so both of these things, if we increase kcat or lower km, will cause this whole thing to go up. And so the opposite is also the case, where if our km goes up, if we're binding less tight, or if our kcat goes down, if our turnover rate, if our actual catalysis is going down, then our efficiency is going to go down. And so one thing I wanted to point out, this is often kind of overlooked in enzyme kinetics, is this and I touched on it very briefly in an earlier video, is this idea of orbital steering. And this is one of the things, I mean, it's probably the main thing that controls the catalysis step, that controls this K2 up here, this K2, which is kcat. K2 is the same as kcat. And so the thing that controls that is this thing called orbital steering. And that has to do with the way that your substrate is aligned inside the active site. And so we might have a substrate, and let's say it has an orbital coming off here. It has some kind of empty orbital. Maybe we have some other molecule, and this could be maybe a residue coming off the protein itself, or it could be some other substrate molecule, and that has some kind of orbital coming off. And so if we have, so maybe this one is an orbital so this is our nucleophile, it has this lone pair, and say maybe this has this empty orbital. And if we are like this, where it's this 90-degree turn, then you're going to get no catalysis. This will not form a bond. And then maybe say instead we're like this, where they're nice and lined up, then this is going to form a bond with that. And we can see that here. So this is the angle in degrees. So somewhere, so 18 to 0 degrees. So I showed it at 19 degrees. So we can actually see that maybe at 18 degrees it's something more like this. And we can see even this small change in the angle of our orbitals reduces the rate of our catalysis considerably. And so we can see when they're at 0 degrees, which I guess you could say is sort of the same as 180 degrees, but they're nice and lined up, they're lined up perfectly, then we get this, then we get our maximum rate. And so our kcat would be the highest that it could go, so it would be multiplied by this 1. Whereas say if we're at 12 degrees on this B line here, if you're interested in this, I have several links in the description about about orbital steering. But let's just say we're on the B line here and we're at 12 degrees. Now we have to multiply our kcat by, something like 0.005. And so that's going to reduce our kcat considerably just because we have this small change in the angle of our orbitals. And so orbital steering inside the active site is a huge part of what determines this right here, the kcat. It's not so much for the km, which has more to do with sort of everything else here in the active site, all these sorts of bonds going on here between amino acids making up the walls of the active site and your substrate. So the way that they fit in there. So for instance, we can look down here at this chirality. This fits in here nice, so it would probably have a very low Km. But now if we have this steric hindrance here, our Km is going to go up precipitously because this is going to make it so that this enantiomer is not going to fit into the active site as well. And so those are the things that determine our kcat and km. Another thing that determines it is actually the diffusion of our substrate and our enzyme in solution. So for the most part, if we look at our Fick's law here, so this is a concentration gradient. If you know your calculus, you'll recognize that as the gradient right there. And so that's if we add a drop into, say, this, looking down into a container of liquid, we add a drop and it's going to start diffusing out. And so we will at first have a gradient around here where the gradient is going up towards there. And that's why they put the negative there so that it ends up flipping the gradient. But anyway, and so this is talking about the flux of our, say, our substrate, and flux is just the amount of it moving through a given area over a certain time. So it would have units that would be something like concentration over area time or something like that. And so down here we show what our D is, and that is based on this thing called the friction coefficient, which I forget the guy's name. I can't think of it offhand, but that can be calculated as this product of the solvent viscosity and the solute radius. So if we just model our substrate as a sphere, and then we just get the radius of that for this, and solvent viscosity is sort of how slimy your liquid is. So something like water has a somewhat low viscosity compared to, say, maple syrup. And anyway, so if the radius of our substrate goes up, or if the viscosity goes up, then our constant will go down, and so our flux will reduce, and so that will reduce the diffusion rate. So I found this from some website. So this is the diffusion rate in water. So this is the sort of average diffusion rate in water. Um, and so we can see that well, actually, if we go over here, this is from a paper, and I think I have that one in the description down below. But anyway, we can see here that for most uh, most enzymes, so this is the kcat over km, that's that catalytic efficiency. Uh, we see that this 10 to the 9 here, which is this over here, once we get into that range, then we are limited by diffusion. So these would be enzymes that are so fast that the rate at which their substrate diffuses in water is too slow. So our enzyme here, if we have substrates, so we might have a substrate come in, and then it catalyzes and forms product. Then if we move through time, even if our substrate is extremely concentrated, we will end up at a point, we will end up with very, very brief, but moments of time where our enzyme is empty, that it does not have any substrate in it because another substrate here has not had time to diffuse into it. And so these enzymes here are examples of enzymes that are that are sort of limited by the diffusion rate. And so we see things, acetylcholinesterase, you might have heard of that. If you're into things like nutrition and nootropics, you might have heard of this as a kind of nootropic. You might have heard of acetylcholinesterase inhibitors. So acetylcholinesterase is a pretty fast one. It's not yet quite diffusion limited, but if we go down to SOD, which is superoxide dismutase, and so this is an antioxidant enzyme, and of course we want that one to be super fast because when we have superoxides, which, if I remember correctly, are oxygens that have a free radical on them, and these are extremely reactive. And if you just have these floating around in your cell, they're just going to start reacting with everything and causing all kinds of havoc. And so you want your superoxide dismutase to be extremely fast because you want to get rid of this as soon as possible. And so that has evolved to be extremely fast, so fast that it's limited by the rate of diffusion. So essentially, as soon as this type of species is generated, that enzyme is already eating it up. And so that's why you would want an enzyme that fast. Rubisco up here, we won't talk about that much in our videos on nutrition and metabolism because this is a plant enzyme, and it has the distinction of being the most abundant enzyme in the world. It is the enzyme that plants use to fix carbon dioxide. So when plants are breathing carbon dioxide, so then this is the enzyme that it's actually using to fix this carbon dioxide. And so it's called Rubisco, which is short for ribulose 1,5-bisphosphate carboxylase oxygenase. And it's called carboxylase because of this, but it's also called oxygenase because it can also do oxygen, which is it's not favorable because that then competes with carbon dioxide and slows it down. But that's all I'm going to say about Rubisco. It's an extremely important and the most widely abundant protein in the world, but it doesn't figure into our nutrition and metabolism that much. And so down here, I just wanted to point out some ideas with the Km and the V max, which, as we know, is equivalent to our kcat. And so, you might think, well, why wouldn't we just evolve for, every enzyme ever to have as low of a Km as possible and as high of a kcat as possible? And there are sort of physical reasons. I mean, for instance, Rubisco is sort of uh, slow, and there seems to be sort of physical reasons, sort of physics related reasons why that can't get faster than what it is. There are biochemists working on trying to engineer Rubisco that's faster as a way to help plants grow faster and maybe even remove carbon dioxide from the atmosphere. But anyway, there might be physical reasons why certain enzymes can't get faster, just because maybe their substrate uh, is such that it can't get any faster. But there are regulatory reasons why too, and sort of the canonical example is the glucokinase and hexokinase. And so glucokinase is found in the liver, and hexokinase is found in muscle. Muscle I think is the big one. Uh, the brain as well, and so hexokinase. I guess I should talk about what hexo and glucokinase are. So these are the very first enzymes in the glycolysis pathway, which is what turns glucose into energy. And so this is the first enzyme, the first step in the pathway of breaking down glucose and turning it into energy. And so if we're at our normal blood glucose, then we want our hexokinase, which is what's feeding our muscles and our brain and our sort of everyday physiological activities. We want that to be uh, at its max, and so we want that to have a a a km that's very low. So we want it to be hitting its max at a very low concentration, because when we're at our sort of normal fasting blood glucose concentrations, we want that glucose going to our muscles and to our brains, where it's going to be used for, you know, staying alive. But if we eat and our blood glucose concentration goes up, so if we eat and say we're here before we eat, then we eat some sugary food and our blood glucose concentration goes up. Now what we want is to store that glucose in our liver, and so what we want is for our glucokinase, which is the form of this enzyme. So these enzymes are similar. They do the same thing. They just do it a little bit differently so that they have different KMs and KCATs. So if we have our blood sugar up here, now we want this to be sort of doing a lot of the work, because we want to try and remove that glucose from our blood so that we go back to our normal blood glucose levels. And so we see here that that this reaches its max pretty quick. So this, which has this higher Km, reaches its max much slower, but it has a much higher max. And so we see that if our blood glucose goes up, then we are here, and this reaction is sort of winning out over this reaction. And so we're drawing a lot of that glucose out of our blood and storing it in the liver until we get back here, where this is, if we're at this concentration, this is happening slower, and so this right here is what's winning out. So we're not storing glucose when we need it to live and function. And so that's just that's sort of the canonical way that's often talked about of how the differences in Km and kcat can be used in different parts of the body in order to regulate how different molecules, in this case glucose, is being metabolized. So this says glucose sensor, and you can kind of see how that's a sensor where this is very responsive to different concentrations, and so it's sort of like it's sensing the concentrations, where this just kind of goes right to its max right away, where we're pretty much always working at the highest capacity here. So the liver, for instance, will store that glucose, and it's pulling glucose out of the blood until we get here. And then, so this doesn't just eat up all the glucose again, then the liver starts actually slowly releasing a little bit of glucose back into the blood all the time. All right, so let's get back to here. So when we get our Michaelis-Menten equation, which we can get if we know the Vmax and Km, then we can plot it using as a function of enzyme, or as a substrate concentration. Like I was saying before, when we do our steady state kinetics assays, it's our Vmax and Km we're looking for, so that then we can fit the curve. We can fit this curve to our equation. But then we can take our equation and we can, we essentially just invert it. So we see over here we have V equals Vmax times substrate concentration over Km plus substrate concentration. Over here we just take the reciprocal of everything, and then we end up with this. And so this is our y-intercept, and this is our slope, and this is our x-intercept. So from this we can do what's called the Lineweaver-Burke plot. And what's nice about that is you plot it here, and we have 1 over Vmax at the y-intercept and 1 over Km at the x-intercept, so negative 1 over Km. So if you want to get your Vmax, you just divide 1 by this, and if you want your Km, just divide negative 1 by this, and that gives you, it says right here, to get your Vmax and your Km. And so we can actually, what's helpful for that is thinking about what's called inhibitors and the different kinds of inhibitors. And so we can have what's called a competitive inhibitor and a non-competitive inhibitor, and there's actually over here what's called an uncompetitive inhibitor. I haven't really run into that very much, so I'm not going to talk about it too much, but what we see here is that if we're using a competitive inhibitor, then we have the same Vmax. These will this will eventually end up at the same V max. I didn't draw that very well, but we end up at the same V max, but the Km is well, it's -1 over that, so the Km is actually higher, and so that means it's binding less tightly, which it should make sense because a competitive inhibitor is if we have our enzyme here, then this is our active site. Our substrate wants to come down to the active site, but if we have some kind of inhibitor there, it's in the way. So this is not binding to our enzyme as much, and so that's going to increase the Km. On the other hand, if we have a non-competitive inhibitor, then our Km will be the same, but our Vmax will be different. And so we see here we max out lower than what we have with no inhibitor, but our Km's are the same. And so you can think about this if we have our enzyme here, we have our substrate up here wants to come down here, but this is not competitive, so it might be bound over here, and substrate can still bind here, but this inhibitor is just stopping it from actually going from the substrate bound enzyme to products, and so this is being slowed down, whereas in competitive, so we had our E plus S goes to ES goes to product. So when we have a competitive inhibitor, we're inhibiting this. So this is comp, and if we have a non-competitive non-comp, then we are inhibiting this part here. And I actually have, well, so first let's say, just mention this. These are how the Michaelis-Menten equations are changed for a competitive, for a non-competitive, and an uncompetitive inhibitor, if we have some kind of inhibitor present. And using inhibitors is a way, so that's used in science just for doing kinetics assays. Sometimes you have an enzyme that just works too fast, and so you might want to compete with it in order to slow it down. But then you can use these equations here to sort of calculate what your V is anyway, given that you know what your concentration of inhibitor, which is what this is here, and you know what your inhibitor bonding affinity is. But we can also see this in biochemistry, because as I was talking about earlier on, when so when we have the allosteric effects, so I was talking about that before, where if we have an enzyme, we might it might be taking substrate and turning it into product, and then product might be coming back and actually inhibiting this in a negative feedback loop. So this is just something you'll have to get used to once we start talking a lot more about pathways and inhibition. When you have something going to something, you use an arrow. When it's inhibiting something, you use a flat line like that. So the product is actually inhibiting this reaction. So if our product is going off and being used by something else, it's not going to be inhibiting this, so we'll keep turning substrate into product. But if for whatever reason the product is no longer going to that, then we will build up our product concentration, and that will inhibit our enzyme. And so that's a way that you can just right off hand that I can think of where inhibitors are used in biochemistry. And so here is basically just kind of what I've already talked about and diagram before. So we have the non-competitive inhibition, which so if we have something bound to it that stops the catalysis from happening, then we have inhibited it. Competitive, our inhibitor will actually fit into the binding site and prevent our substrate from entering it. And end product inhibition is what I was just talking about, where we turned this substrate into product, and then the product comes and binds there, and this becomes inhibited. All right, changing gears a little bit, I am now going to briefly discuss receptor kinetics, which it has a lot of sort of similar ideas to enzyme kinetics. So in receptor kinetics, we have, so instead of calling it substrate, we usually call it ligand plus receptor, and that goes to ligand receptor. But in this, we do not have a catalysis step. This, all we have is this, is we want our ligand to bind and unbind to our receptor. And so for that, we have something similar to Km, which is Kd, which is when we are at half capacity. So that's when half of our receptor is free. It does not have a ligand bound to it, and the other half is ligand bound to receptor. And the Kd is actually calculating the off rate. So it's actually calculating that right there, this back reaction here. So you will often see it written as this, which should look familiar from our equilibrium constant, except if we were doing equilibrium, actually it's supposed to be uppercase K-E Q, you would probably see it written the other way, because this would be talking about the binding of the ligand to the receptor. And I mean, this is a thing. I think they actually, forget what it's called. It might be KA, something like that. But this is a thing. But in when you're doing receptor kinetics, you're pretty much always using KD, which is so it would be this, and KD is when we are at half capacity, and so that would be so yeah, so the KD is a concentration, so that's a concentration, but it's the concentration when we are at half capacity there. And so just like with Km, if we have a lower Kd and we have this higher affinity, which makes sense because if we're looking at L R over ligand bound to receptor, so lower Kd will be if this goes down or if this goes up. And so if we have more of this, then that means that our ligand is bound to the receptor. And so if our denominator increases, then we are increasing the affinity for it. And sort of the opposite, if we have L, R, and L, R, if we now have the denominator going down and this going up, then our KD will be higher, but it will be lower affinity. And part of me thinks that's why they do the KD rather than this KEQ or KA or whatever this is just so that this is the same as it is for the KM. Because if you had to go back and forth and remember which one it is, that if it's lower, then it's tighter, and one if it's higher, then it's tighter, then it would just be confusing. So I think part of me wants to say, I don't know for sure, but part of me wants to say that's why they do it this way, so that we can just be less confused. And so down here I just have some instances of receptors where a ligand is binding, and you can see on this that it's causing this conformational shift. We have this here that's shifting when this ligand binds. I really like this picture because they have the amino acids that bind to the ligand here. And you can see them sort of like almost like a mouth sort of biting down on this glutamine. And so, like I said, this is not a catalytic reaction. This is just a ligand bound to a receptor. And what happens is this generates a conformational shift. You can kind of see that this loop is different here. Maybe what happens, I don't know for sure, but what is often the case is, say this now presents this this part of the protein, and now some other protein or something can come and associate with it, and that activates this protein to maybe catalyze some reaction when it's bound to this, or things like that. That you see that a lot in when we're talking about receptors and signal transduction. So that's sort of where this receptor thing is going in later videos, is signal transduction, which, just to give a quick preview, is we have a ligand. Quite often there are receptors that are not, but most of the time our receptor will be in the cell membrane. So this is the cell membrane, and part of it will be sticking out of the cell. So this is outside the cell and this is inside the cell, and the ligand will bind to it. That'll cause some conformational change that sort of propagates through the protein. Maybe it opens something up right here so that some other protein will bond to it, and then that protein will start. Usually it is it's phosphorylations, so you'll end up with your substrate with phosphates on it, which uses ATP and produces ADP. And so that's we'll, in later videos we will see this type of thing quite a bit. And so that's sort of the relevance of these receptor kinetics. I think I'll quickly go through this. So this is just going through ways that you can actually measure the receptor kinetics and try and find that KD. So there are different ways using fluorescent protein. Well, fluorophores of some kind. Sometimes it's a fluorescent protein. Sometimes it's a small molecule that's bound to the substrate. So those are fluorescent. I think that's how it's spelled. But anyway, there are different ways. So you can have your fluorescent molecule bound to your substrate. This right here is the, well, I guess ligand, the ligand. And so this will fluoresce differently when the ligand is bound to the receptor. So that's a way of measuring how much is bound is by how much fluorescence you get. And this is an example of that, where we're increasing the concentration of our ligand here. So these are concentrations in micromoles, and this is our fluorescence here. And so you see at these low concentrations where, say, our fluorescence increases when it binds to the receptor. So over here we might have this low fluorescence, but then once we start saturating our receptor so that all of them are in the receptor ligand bound state, then we even out up here. And you can see, you can get that for, you can put the fluorophore on the receptor instead, and then when the substrate binds, the conformational shift in the receptor will cause the fluorophore to change its fluorescence. And then there is this version, which is called FRET, which is fluorescence resonance energy transfer. And so that's what happens is you're shining your light on your solution, and the light would hit this and it would fluoresce as red, and on this there is no fluorescence going on. However, when you're hitting this with your light and it fluoresces red, then that red light goes to this and causes this to fluoresce green. And so only when these are in close proximity to each other will you see the green fluorescence. That's what I have down here, is this FRET. Now, I'm not going to go too much into this because this video is already getting quite long. But you see here you can have you can do FRET for a single protein. So if your protein has some sort of conformational change that brings these together, you can measure that using FRET, and you can also do it with two different proteins. So you can have your proteins come together, and now the light you're shining on it maybe is hitting this one, and then that releases a photon that goes to this one, which then releases a photon that's blue. So this releases a yellow photon, this releases a blue photon, and now this releases a blue photon only if it's getting hit with a yellow. Say what you're using here is maybe red light or something like that. So this doesn't fluoresce when it gets hit with red light, only when it gets hit with the yellow light. So when they get close together, this will fluoresce, and you can detect that. And you can see that here we have our when our molecules are close together, we hit it with a light, it fluoresces, which excites this one, and then that releases fluorescence light. And when these get too far away from each other, then we lose our fluorescence efficiency. I'm not going to go through the math here. If you're interested, it's just on the FRET Wikipedia, the Wikipedia about FRET. So if you're interested in that kind of stuff, you can go there. This is just another common technique used for determining the KD. So we're still looking for our KD of our receptor ligand system. And so this is called surface plasmon resonance. I'm not going to go too much into it. You can look that up online too. I I just think it's a really interesting kind of technique where you're shining this light on the surface, and you get so this shows it as antibodies, but you could also have ligand and receptor as well. So you get evanescent waves that change the conductance of the surface, and that changes where the absorbance and reflection happens, and so you can measure that difference there. And this is the difference. So what you actually do in here, instead of just having this sitting in solution, you're actually pumping solution with a constant concentration of your ligand through, and so you're constantly getting the on and then off, and then when it goes off, it goes away. But when you have new coming on, so yeah, that's these fluorescence and surface plasmon resonance. These are not the only ways of doing receptor kinetics, but they are common ways of doing it. Oh, this video is getting a little long. I'll quickly go through. I want to just look at some proteins here. So this is actually the coronavirus, the receptor binding domain down here from COVID-19, from SARS-CoV-2, and we see the red here is a binding domain. So this is binding onto this ACE2 domain on the human lung protein, on this human lung receptor. So this is a receptor in the human lung cells, and you can see that this the COVID-19. So this is from its spike protein. This is just the domain, what's called the receptor binding domain, and that's binding to the human ACE2 receptor. And I colored this red here. This is where it's making contacts based on the paper that I got this from, which is linked to in the description below. So I colored the place that's making contacts with the human ACE2 here in green. So that's just a way that we can see receptors binding to each other. That's sort of it's sort of relevant to current news as I'm recording this video. So that's just I thought was a interesting instance of binding to a receptor. Then I have this one, which so this is a so here in the blue is a retinoid receptor, and we'll actually run into this more later on when we start talking about vitamin A, because vitamin A, so this is a retinoid here. Vitamin A is also a retinoid, so those act as ligands for retinoid receptors. Then there's this PPAR. I don't remember exactly what that stands for. And this has, I think this is actually, I don't remember if this was an inhibitor here or an agonist, which is, an agonist is something that's just, it's not its usual substrate or ligand, but it's something that can act like it. And so we see here, this is a receptor. This retinoid receptor is a receptor, and it has this ligand bound here. This has sort of a ligand analog bound to its receptor binding site. These little green guys here are they're they're nuclear receptor proteins. So when these have their substrate bound, that causes a conformational change in the protein that allows these things to bind. And then what that does is these, which have these I don't know if, I think this shows up. So you can see up here the sequence for it on the top there, and it has this LHRLL. That's the LXXLL motif. L, then two whatever amino acids, and then two more leucines, and that is a little protein that gives this access to the nucleus. So it's usually in the cytoplasm, sitting around waiting for its ligand to bind. When its ligand binds, there is a conformational change that allows this to associate with it, which then allows it to go into the nucleus, and then it comes down here and binds to the DNA. So you can see the DNA structure here, and you can see these things going into the major groove of the DNA, and this one has one that goes into the major groove. And you can see these little balls here are zinc ions, and so these are sometimes called zinc fingers because they have zinc and they're kind of like little fingers poking into the DNA here. And so you can see when the ligand binds, this nuclear receptor protein can associate with it, which gives it access to the nucleus where the DNA is. It binds to the DNA, and this acts as a transcription factor, which I talked about in the video on DNA and RNA, and we'll be talking about more in later videos. So it's a transcription factor. So this will, for instance, associate with the giant mediator complex, which then recruits RNA polymerase to specific genes based on where this is bound to the enhancer. And so that's just another example of receptor binding. And I think that's everything I wanted to talk about, and even if it's not, this video is already quite long, so I will leave it there, and I will see you in the next video.